Hi class, today we're going to be looking at meiosis, which is chapter 8, section 3. Meiosis occurs when we need to make egg or sperm cells in order to make gametes. That's what they're called. And the purpose is so that we have egg and sperm cells that are made with 23 chromosomes. And when they combine to make a zygote, we get 46 chromosomes. So meiosis has two stages, and you can see if you're looking at your notes packet that these stages are written out for you, and then we'll go through some of the things that happen in each of these phases. So first we have interphase, which is similar to what happened in mitosis. There's three stages, G1, S, and G2, which were all the same as what we saw before. G1 is a growth phase, S is a synthesis phase to make a copy of DNA, and G2 is a grow 2 phase where it just gets ready for mitosis. In this case, it gets ready for meiosis. Meiosis 1 is when we form two haploid cells from one diploid cell. So in this case, we are going to go from our normal diploid cell to creating two individual cells, two daughter cells you'd call them, but they're haploid. That means they have half the number of chromosomes. And this is going to occur for the production of gametes, and it's called reduction division. So prophase 1 is when we see the nuclear membrane break down, the centrioles will move to the poles, and the spindle fibers will form, and the synapses will occur, forming tetrads, allowing for crossing over. So all of these things here above this point are all things that we've seen happen before. This next part here is what we see happen in meiosis compared to mitosis. We didn't have this process down here of the synapses happening in mitosis. So I'll give you a couple definitions of what that the synapses and tetrads and crossing over is. Basically, crossing over just allows for genetic variety. Otherwise, we would have so many similarities to our parents or to even our siblings, where when we have crossing over, it allows for some extra variability among individuals. And you can see here basically what happens is we have these chromosomes that line up, and when they line up next to each other, like this here, when they line up next to each other, their legs cross, and what happens is they kind of twist together, and we see that they switch that leg of the chromosome, and that allows for some variation from the parents. So to fill in your notes packet, here's some definitions. A synapsis is when a pair of homologous chromosomes partner up inside a dividing cell. So they're homologous, meaning you have this equal pairs, chromosome 1 next to chromosome 1, chromosome 2 next to chromosome 2. And tetrads is what we call that, when the homologous chromosomes line up next to each other, because it looks like a group of 4. And crossing over is when we see that leg of the chromosome, they kind of twist around each other and then trade places. And so it exchanges that DNA. So instead of just getting the straight chromosome 1 from your mom, you're actually getting a combination of what was chromosome 1 with maybe just a change in the leg, which came from the other chromosome, which was actually from their dad as well. So they're getting some combinations that didn't come from just one parent. It's allowing for variety. This is really important for organisms out in the wild because it allows them to have variety in their offspring that maybe they wouldn't have had otherwise. Okay, step 2 is metaphase 1. And in this, the tetrads line up randomly at the metaphase plate. So you can see that happening here. Again, metaphase is always where they line up across the center of a cell. Anaphase 1, the tetrads are split and each homologue is moved towards opposite poles. And you can see that happening here in that picture. This is where another interesting thing that allows for variety happens. Independent assortment is basically when it's a random separation of those chromosomes. And when it's a random separation, you get variations in your offspring. So not everyone is going to look alike, because it doesn't mean that all of these same chromosomes always get set together in a cell. Instead, you're going to get a variety of the chromosomes going to each cell, and it's going to change up how the offspring look. Telophase 1, the nuclear membrane reforms, the spindle fibers and the centrioles disappear, and there's 2 nuclei per cell. At this point, we call it a haploid number of chromosomes. Now, cytokinesis 1 occurs at this point, and this is when the cytoplasm produces the 2 daughter cells. Now, meiosis 2, if you go back in your notes packet, you can fill in what meiosis 2 occurs or how that occurs. There's no interphase between meiosis 1 and 2. So there's no extra growing, there's no synthesis of DNA, none of those things happen. So what's going to happen now is we're going to produce 4 haploid cells from those 2 haploid cells that we just had. And basically, it's the exact same process as mitosis. Prophase 2, metaphase 2, the chromosomes line up across the center of the cell. Anaphase 2, they get pulled to the opposite ends of the cell. Telophase 2, they start that cleavage furrow or cell plate. And then cytokinesis 2, the end result is 4 haploid cells. You can see that here. These 4 haploid cells are called gametes, and that word you will see come up again. It's something that you need to make sure you know goes with meiosis, and that's the egg and the sperm cells. This is a picture showing that again, interphase for meiosis 1, interphase, prophase, metaphase, anaphase, and telophase 1 with cytokinesis with that cleavage furrow there. And then meiosis 2, same process, but it's with 2 cells occurring at the same time. And then in the end, we get those 4 cells. So the purpose. We have gametes, which are sex cells. They're the egg and the sperm. And there's 2 names for this production of gametes. So oogenesis is egg production, and in egg production there is only 1 good cell, 1 good egg. And the other 3 are called polar bodies. They actually don't function. They are not an egg. They don't have the DNA. They have the DNA. I guess I should take that back. They do have DNA, but they don't have all of the other structures. They never grow enough to become an egg. So they don't get all of the other things that the cell would need to be able to survive. Spermatogenesis is sperm production, and it does produce 4 good sperm, 4 viable sperm. And then sexual reproduction is just genetic diversity of offspring. So we're getting a combination of information from the mom and dad to produce offspring. And it is better for offspring to be reproduced by sexual reproduction for the fact that there's more variability among individuals, and that in the environment for organisms that are out in the environment is very useful when environmental changes occur or when there's situations out there that make certain traits to be more favorable. Okay, this just shows again spermatogenesis begins in puberty for boys. You can see there that in the sperm we get 4 viable eggs, or 4 viable sperm, sorry. Oogenesis occurs before a female is born, and there's only 1 viable egg out of the division. In sexual reproduction, each parent contributes genes to the offspring, and each offspring has a different set of inherited traits from the parents. So you're not an exact copy in any way to your siblings. Now, identical twins do have similar properties, and they do have similar genetics, and they are produced from the same egg and sperm if they're identical twins. So that is a little bit of an exception. Basically, this idea of sexual reproduction does produce better chance of surviving in a changing environment. So where there's genetic variation, it's helpful for organisms, especially out in the environment, out in the elements, that they can have that variety so that their species continues to survive. And here's showing that again a little bit with our crossing over and some of that variation. Okay, we're going to end there, and we'll do our comparison of mitosis and meiosis on the next set of notes.